Again, spread the word. This is an all-volunteer effort. It's all about getting people from the community and having scientists speak about subjects of general interest related to their work. And so we're very lucky to have Heather Bradshaw join us. Heather has been with us with Bloomington campus for probably 7 or 8 years at this point. She's faculty in psychological and brain sciences department. And she's a fellow cannabinoid researcher. She does a lot of different things. And Heather's a very dynamic speaker. She's great. She's a great speaker.
Let's hope.
And this may be the case this time as well. No, but she's the only person to whom I have, because usually when I go to someone and say, we've got an idea for a science cafe, we need to do this, she's the only person whom I said, you can talk about anything you want. And
really?
Yeah, absolutely. So we have faith. Yes, I think this will be highly interesting for us. But so Heather Bradshaw.
Thank you. All right. So as Alex said, because one of the courses that I teach is try to stay out of the view here, especially since a lot of them are visual things, is Introduction to Brain Sciences, Psychological Brain Sciences. And so that's a really broad field. And so I actually came from a biology, nutrition, and physiology background and didn't really study psychology. And so when I started the psychology department, I really had fun actually learning more about general neuroscience as a whole. And so the title is in reference to the fact that through teaching these courses and through working in signaling systems, which is what I do, that understanding that how our cells communicate is one level of how our body works, but how our cells communicate with the world, with the energy of the world, with the chemicals we interact with the world, is the underpinnings of how we actually perceive the world, meaning how we think and feel about the world, and that it is very different person to person because of all of these differences in how these energies can get into our bodies. So the question that we have to think about is not really a philosophical question, but it's more of a nuts and bolts thing, but it's a definitional thing to think about. So how do we sense and perceive our world? They're actually two very different things, and I'm going to kind of walk through some examples of what that means. When I talk about sensation, I'll just read off, the process by which our sensory receptors transduce stimuli from our environment to our nervous system. So I'll break that down. So, sensory receptors, right? So these are these specialized proteins, organelles, systems that we have evolved to be able to capture energy, different kinds of energies from our world, like photons or airwaves or chemicals, and to transduce something. Does anybody know what it means to transduce something in a general sense? Just throw it out there. It's a really simple word. It's like to change from one source of type of energy to another, to change something, to transduce something. And we actually can do that. It is so cool. We can capture a photon and transduce it into a neuronal impulse, into a neuron firing. We have that ability. Any creature that is able to, in any way, see can do this, and it's really awesome. So, but that capturing, that ability to do that transduction is actually sensation. And one of the things we as humans, and we think a lot of other organ systems as well, we can talk about it as humans, we have this other dimension called perception, and that is actually the organization and interpretation of the transduced sensory stimuli into meaningful information. Okay, so you get this information coming in from this environment, but what does it mean? Does it have any meaning? And that's what our brains do for us. And that is really this amazing thing that just happens And we don't always know exactly how that happens. And it is an incredibly complex system. But it's fun, and we're going to experience basic sensation perception tonight. So, some of you may have seen this picture before. If you have, don't shout out yet what it is. What you're doing, if you don't know what this is, you're sensing. Can you see this next? Do you know what it is? Okay, so everybody is experiencing basic visual sensation where you are able to bring these photons in, transduce them enough to say, okay, there's some black and some white, right? So, who knows what this is? What is it?
It's a cow.
It's a cow. Welcome to perception. Now who sees it's a cow? Snout, nose, eye, ear. But how cool is that, right? You see? I mean, this is like a fence. You see that? You get it? She's like, so, right. So this is the beauty of this whole concept of how our brain works to put together information and put things into meaningful units, right? We just as a group experienced the fact that we equally were able to bring that information in, but until we had it in a context, we couldn't give it meaning. Now, I don't have time to go into the difference in what we call bottom-up processing, which is what you did at the beginning, and top-down, once somebody told you it was a cow, okay? But that's part of this whole perception and how our brains work to integrate sensory information. So, we'll play along and have a few more. There's another animal picture. So we see the foxes. What other animals do we see?
Humans.
Yes, you see the people. Who doesn't see the people? You don't see the people?
I don't see any people.
You don't see the
Aww.
people? This is one person. You see that? Another person.
Oh, okay, nice.
Yeah, yes. This one's kind of broadcast, but I think that, your first, you've also been primed now. That was top-down processing. You knew to look for something that you didn't think was going to be there or wouldn't be obvious, right? And so, but because we have a context, our brain is helping us. It's top-down processing. But everybody's getting that same visual information in. Some of the earliest work on this used this type of picture. And probably most of you have seen this. So what do we see in this picture? We see two things, right? What do we see?
Is there a vase or two faces?
It's either a vase or two faces. Now, the fun thing is that there's a couple of things. Is that you had to know the context. You had to know what a vase was. You had to have that in your cultural, you had to have that in your sort of mental schema toolbox. To know it's a vase. Faces are easier to pick out, the contours. However, you have actually got to switch back and forth in your head constantly to either choose to see it as a face or a set of faces. It's practically impossible to see it and cognitively understand it as both at the same time because our brains want to choose. We want to categorize, we want to say, I see this thing and this has this meaning. And I love seeing your faces because everybody is trying to do it. Everybody's like, no, I can do it. No, wait. No, wait. It's not a face. It's a face. No, yeah. Okay. Another classic picture of this from around the turn of the century. Some, I believe, German scientists came up with this great picture. And it's also, not only is it, so again, it's a very famous picture. So who's seen this? A couple of you. So it's either a young woman or an old woman. And again, it's even more ever see them both at the same time, your brain keeps switching back and forth. You see the young woman, this is her jawline, say a necklace, her eyelashes, right? And again, I say the bottom of the time because this is more of a late 1800s, early 1900s sort of look for a woman. And then the old woman, this is her mouth, her chin, and say she's wearing a covering, right? You got it? Yeah, I see the note. Oh, yeah. But the beauty is, when you look and you see the old woman, this is what your brain tells you that picture is and what that meaning is. And then it has to switch to see the young woman. So we have a perceptual switch that we allow ourselves to experience. When we decide what context we're going to be thinking about the information coming into our brains. So, this is one level of how our brain works. But to be fair, especially with black and white, except in very extreme cases, right? Everybody's going to see this black and white that can see, that has visual perception. But that is not the whole story, of course, of the senses of perception. Is something as base as that. We've all heard of the five senses. We get this from beginnings of school. What are they? What are they? Shout them out.
Vision.
Taste. Smell. Touch. Feeling.
Vision.
Hearing. Sight. Vision. Yes. So, what are they? Vision, hearing, smell, taste, touch. These are categories. But again, going back to what do we actually sense? Are those more this sort of organizational units of all these things that our brain is doing for us? We actually sense things like photons and air pressure waves and chemicals and mechanical pressure and thermal differences. This is what we sense. This is how we interact. This is how this information comes into our bodies through very specialized receptors just for these actual physical components of the environment. And so we experience being able to get photons coming in and understand that we also have differences in perception of that. But the fact is that there are ranges of how much of each of these different kinds of receptors, specialized receptors, that individuals actually make. And this leads us to bringing in different amounts of information and therefore having a very different perceptual experience. One of the classics to consider is about light. And what is that? What is light? This is the example of light being shown into a prism, and you see this range of colors. Why do we see that? Where they're actually, the question is more aptly than what are the photons that we are capturing, we can see. And again, incredibly simplistically, which is, I know we have a physics person here, photons are particles that also have a wavelength and an amplitude and a frequency. And depending on that amplitude and frequency, we will be able to capture these different wavelengths into, we can only capture this little range here. Our specialized receptors can capture here, and we then label that, most of we will show you that that is different for different people, we call it color. Color would be our perception. Okay? So the photons coming in get captured by different specialized cells, but only we only capture this very, very narrow range. There are other organisms that can capture other ranges. Their world is very different from ours perceptually. And we humans don't all get to capture this range of wavelengths either. So, we have the eye, which I won't go too much into, a little square back here, giving us a representation of these specialized receptors that are called the rods and the cones. The cones are what are the specialized receptors that have different proteins that can capture these photons at different wavelengths of photons. And there are only 3, generally, that we know about. It seems very narrow that we would only have 3 and be able to see such a rich world as most of us do. But of course, it's never that simple, and we've evolved really cool ways of giving us more information from the same bits of information. And one way is to give some sort of dual weight to how these receptors work when you're getting a wavelength into your eye. Now, this is the fun stuff, part of the fun stuff. We're gonna all experience together something called a negative after image. I don't think it's negative in that it's unpleasant. It's actually pretty fun. So, the deal is, and some of you may have done this before, but I still get mesmerized when I work on this lecture again, and I just keep doing this over and over again. Stare at the dot. Stare at the dot really hard. And what you're doing is you're allowing this sensory information to come in and basically some of your photon, your cones, your specialty receptors. And now keep looking at the screen. And now what do you see?
Flag in the right.
Yeah. Red, white, and blue flag. For those of you who can see red. Okay. So that's kind of fun. And who wasn't able to see it? Maybe your angles. No. We'll do something else. We'll work on a different section. We'll work on our rods. I like to look at Batman's chin. You just focus on it for about 10 or 15 seconds. And that is why I brought the line up. Okay. So keep looking at the screen. And does Batman fly away a little bit? Around. So this is fun. So what you're doing, yes.
Is it moving away because of our skein? Because we tried to follow it?
So Tasha's question was that, see, our eyes are always moving. And that's something else. This is, we're going to see a few more of these. So we'll look at this picture. And the interesting thing, too, is this one is incredibly effective, I think, for multiple reasons. Keep staring at it. You know what color it's supposed to be, right? You know what color you would suspect this to be. You're still staring at it. Give yourself like 4, 3, 2, 1. Okay. Now look here. Like blink once. How red is that getting? You see that? Nobody see that? You can't see that in the back? Is this working? Yeah. So now, last one, we go back to the rods and the cones. And for me, I often see, just stare at this again. We'll get to see even more. Really bright, typically. And sometimes the red after image is still there for some people. So you have like the Canadian flag with a light bulb. It's not propaganda. It's just what it is. So we have not a very large group of people here tonight. However, there are some people that would not be able to see and have that same experience that we all just had, and experience, especially color red. Many of you have taken, I don't know, does Indiana do this for their driving exam? I don't remember this. Apparently, it's illegal to drive if you are color deficient in Romania. They will not give you a license if you have a color deficiency. This is under, people have been trying to change this law, but everybody typically can see that as a 12. There are individuals who cannot see a number in this. Anybody not able to see a number?
Is it a 68 or 88?
It's a 68. Right. Like I said, I was telling someone before that I teach this large lecture class, and so far I've never not had at least one person that is red color deficient. But not to fear. I have pictures for you of what their world looks like. So, something else that's interesting. So, there are, in this particular blot, those individuals see a 2, where we see a 5.
So if you see a 2, you're color deficient?
You might have some deficiencies. I see a 2 on the screen. I see a 5. I see the 8.
I see.
We'll go back. You're supposed to see a 2 and 5. Like, look again. If you can see the 16, you have no red deficiency, but you might have some other. So here it is. This is what normal looks like to us, to most people who have all the spectrums. Alex, what did you see before? Did you see the 5? You see 2?
I saw a 5, but then I started seeing an 8.
So, one of the situations of not having a set, some of the proteins in your cones that work, this is more what the world would look like to those individuals with the full deficiency of not having the red cones. And it's incredible to think. So here's another deviation on that theme of not having as much ability to capture more in the red range of photons. Imagine we think everybody has the same perceptual world as we do so often, but they do not. There's about over 5% of the population has this, where they don't see these colors. This is a very rare condition of not being able to see yellows and blues, but it exists. This is a great picture, I think, of bringing it back to real world. My oldest brother is red-green colorblind. This is so how he sees an apple. It still blows my mind that exists. That, that is his perceptual world. And that's just how it is. But again, yeah.
Did he learn to recognize that color as red?
You would never know by, yes. You would. I just thought he was a dude and could not dress himself. So, this was no. I have no idea. Like, he says that there's differences. Like, that's the whole point, is that when you develop, and you're told as a kid, that's red, then, yeah, okay, it's like, fine, and that's green, fine. So you say, right, that's red, that's green. But you can categorize it enough. Like, we have all these different shades of gray that we're able to be like, that's a little bit more gray than that. So it's more along those lines, because it's a little more yellow than that. It's a little more. And so, yes.
Wait, what's yellow to these people?
Really yellow. Okay, so we'll switch gears so that we have a demo for to go back into taste. So, again, so then my brother would not not see this as I see this, these red chili peppers. And there's a lot of things that get evoked when people see red chili peppers, especially those who have consumed them, or are afraid to consume them, or salivate when they see this. All of these things because of your perceptual experience with chili peppers. And there are such a wide range of taste perceptions. And again, I think that a lot of us don't realize that that is the case. And I'm going to walk you through what I mean by taste. So, going back to our interaction with our environment, taste, sensation that results when taste buds convey information about the chemical composition of stimulus, food, and drink, we hope, to the brain. And what are our specialty receptors? I don't have really cool micrographs. There are some. They're oval-shaped clusters of cells located on the tongue and the lining of the mouth that transduce and then change chemical energies, chemical information in food and give rise to our perceptions. So what would a perception of taste be, like a global term? Think about it. What is a component of food? It's something that you think about when you think of how something tastes.
Sugar?
Sweet. A flavor, right? How your visual field, how you see something is rich in a color. We think of flavors. Okay, so as a color is a perception, so is a flavor. Okay, and so again, thinking about the difference between the sensory information coming in and what is the perception. They are actually these taste buds with these pores. They're on these little villi all over your tongue. So there's a lot of them, and there are lots of different kinds. Some of them allow calcium to flood into the cell here, which is basically a modified neuron at this point. And some of them start a process that takes a little bit longer for information to go. But they're all over the tongue. And not surprisingly, we humans have an enormous variation on what our tongues look like and how many taste buds and taste receptors that we express. It is astounding. And this is just two examples of a person. They're staying for these clusters of taste buds that can have all these different taste bud pores and everything all in them. And this individual has many fewer than this one. And so then, not only, so this would be, again, like the issue of, do you have the same numbers and types of cones? Do you have the same numbers and types of taste receptors and taste buds? And so, for those of you, I know you're eating, who are interested, I have some things called taste papers. And I will pass them out. Would you like to do this? All right. So what it is, you're going to need to take out. Help me out here. So stand up and walk around and give part of the twos and part of the ones. All right. You have twos and ones there? These two, some twos and ones. All right. It shouldn't take long then. We only have 20. You guys have them yet? You want to do it?
Oh, is yours? Let's see this. I think are we not supposed to get one of each?
You're supposed to get one of each.
One of each?
Yep.
Okay.
So I need it. Oh, I already got
Okay.
Keith and I.
So are you guys doing this? She said no, that's okay. You're all fine? You want to start with you want to?
It won't make your food.
It will not make your food unpalatable. It's just that it's...
A little water.
Okay. So, what you want to do I can pass it out. So, take out, on the ones, there are two different tastemakers, two different colors. And put them on top, and so that you can put them on top of a little white, your little white packet, because they're two different colors, and you want to be able to see the colors. Yeah, but you want to make sure that you don't separate.
You're an optimist.
So, yeah, make sure you put them on the white so you can see. Make sure they go on something white so you can see the difference. Yes, absolutely. You see whites? All right. Anybody not have a scorecard? You guys don't have a scorecard? Everybody that has a taste paper that wants a taste paper has a scorecard. Okay. You need a There you go. All right. Okay. So, what I want you to do, the order that they're on the scorecard is the order that I want you to taste them. So what you'll do is just put one of the taste papers in your mouth and decide if it has any taste, if it's not very offensive, if it's really offensive. You start with the white strip and do a 1 to 5. And then once you've done with the white strip, you can just take a glass and drink of water or whatever, and then do the pink strip. Yep, you can just plow on through them. After the pink strip, and you've made your choice on your number, go to the blue.
These are nasty.
Bradshaw, I got 2 white strips.
Well, you have to look. One's yellow and one's
Not yellow.
not yellow.
I think
Not yellow is blue.
So you have, so the yellow should be in the number 2.
Jesse, right?
Who said that they didn't think they had a yellow strip?
I don't think I go.
control, there's nothing on it. And most people rate it as a 1 or a 2 because you can taste paper. Some people have that. The second one is sodium benzoate, which has a range of tastes. Some people perceive sweet, some people really salty, some people sour. Who felt like pink was more on the sweet side?
I had a sweet and a bitter.
Yeah. How about salty?
Yeah.
I'm a salty for sodium benzoate. I'm totally salty. One? You tasted it. Oh, you didn't taste it at all. Right, so there are people who do not have this receptor. Yeah, who didn't taste it? Nobody, there was a couple people that didn't taste it. And blue is this phenylthiourea, which is this PTC. Who could not taste it? All right. Those of you who could taste the blue one, what was your impression? It was a 4. It was, right? Exactly. So you're like, not so much. How crazy is that? These are people who do not taste it. There are like 6 of them here that don't taste this at all. At all. And you people are going, right? Most people, this is a very low percentage, like 5%. Anybody not able to taste the yellow? Okay.
It took me a little while to taste it, but it was pretty tough.
It's a threshold thing. Most people with enough of it, you can taste this. But this is really crazy to think that we're all eating the same foods, drinking the same wines, beers, sodas, and we have a very, very, very different perception because we have a very different expression of receptors on our tongues. The people that started this work about 20 years ago, I think sort of erroneously, gave the name to those individuals that most people that taste all of them in the extreme have tongues more like this. And those that don't have tongues more like this on average. I do not think it's fair to call those people non-tasters. I think that they taste lots of things. They're just different tasters, right? And but that you should know is the rap that people get that don't express these receptors that aren't able. Yes, Alex. Sorry.
Does this change over time? I mean, do people, can you like kill them off, or can you sensitize them?
What about environment? Everybody says, because there was a mustard commercial 10 or 15 years ago that I loved. These 2 kids sitting on a set of stairs, and this one kid's like, "Oh, you eat that kind of mustard?" He goes, "Yeah, it's an acquired taste." He goes, "How did you acquire it?" He said, "My mother gave it to me." Some things are an acquired taste, but not PTC. We do not seem to, no matter how much bitter stuff you eat, do people seem to ever express this. But going back to our example of chili peppers, there are different receptors called trip receptors that are not classified at all as taste receptors, but they are very abundant in the mouth, and they respond to different things in plants, like capsaicin, which is this lipid in chili peppers. Now, you can make your tongue make less of this receptor by consuming more of the thing that makes it turn on. Such that what happens is that you start to think that your favorite spicy food is no longer spicy. And you think, what are they doing? This place used to have really spicy I eat it all the time. Guess what? You have basically created a tolerance in your mouth for chili peppers. And so you're just helping keep pumping it up and getting more and more spicy peppers to activate what little receptors are left. Yes?
Does that reverse itself when you quit eating the peppers?
It seems to do that. So that is the question. So environment does matter. And so that, exactly, this is the idea that if you stop eating the hot stuff, then you will start to reanimate those receptors.
This goes over to Alex. This sort of reminds me of what you talked about with drugs, that you can eventually start losing your capacity to feel them, so you need more, right?
Yeah, I mean, actually, that's very much the same thing. Yeah, you're developing kind of a tolerance for it.
And is it related, these two?
Yeah, no, I think it's very similar kind of effect.
Yeah.
They affect very, very similar kind of mechanism too.
Yeah. So, yeah, it's tolerance. You're building up tolerance, just like you do with any substance that activates a receptor, that if that receptor is one, if that protein can be, what we say, downregulated, no longer as effective. But that is, yes.
So that's the question, how many of these can be there? Like the sugar receptor, can you get fat because you lost your sugar receptors?
Well, exactly. I mean, this is a great question because I doubt we have any sort of evolutionary need for high fructose corn syrup. But we can taste it. I mean, when you do the taste test after you've not had it, or you go to the UK or something and eat their chocolate and go, wow, why is this so different? Oh, because it's real sugar and not this other stuff. I don't know. This is a great question. Like, do we just desensitize our receptors for things like the simple sugars that would be in a plant that would be much less abundant than concentrated sugars in things that are made with concentrated sugars? Yes.
I'm sorry, this is very curious. So with dogs, for instance, I was wondering if how the senses are comparable. Dogs can be trained to become more sensitive to their smells. Presumably we could as well. Can other senses be trained in the same way?
We can only be trained to a certain extent, and partly because certain animals like dogs and some rodent species, they have almost 10 times more variety of receptors in their nose and their mouth than we do. We humans really don't have that. We only have in the hundreds that we know of certain types of receptors that we know of classes, whereas they can have in the thousands. And so then their discriminatory power is going to be better than ours, just from that degrees of difference, because we make bigger leaps between things. Yeah.
So when we build up tolerance, is it because the receptors are disappearing, or is it because they're just not working as well?
Well, what we understand, is that they're not being produced at the same level and traffic to the membrane. Okay. Because the theory goes just like Mike was suggesting, like the drugs. Any sort of outside, we call ligands, those things that come from the outside and bind receptors we're not making ourselves, exogenous stuff like capsaicin. That there is this feedback that the cell says, well, I don't need so many receptors to detect this anymore. It's not energetically favorable for me to make so many because I can detect that. And so the idea is it kind of backs off. And in that there's a feedback of I need this many receptors to detect this within my environment.
And so the receptors, it's not like you're born with a set of receptors and you keep them your whole life. They're constantly being
Correct. Exactly. They're constantly being recycled. They are, yes, that's exactly what's going on, is that you have this, which is why, too, there is this issue with environment of what do we think is noxious. there are data to suggest that 7 presentations of a novel food, a novel taste, is what it takes to get used to that taste. This is the whole thing of of drinking beer. Somebody said, what's the lightest possible thing that doesn't actually taste like beer? And so that those individuals would, it's degrees of freedom. So the next time you would drink a little bit more of something that's more interesting, to be fair, than the things that don't taste like beer. And gradually your brain stops saying, this is something I should spit out, it's noxious, right? Because we do have that defense mechanism, like this PTC. This is one of these hypotheses that the majority of us, about 70% of us, can detect these bitter compounds way more than others. And the hypothesis being that that is something that would go along with foods that might be poisonous, right? And so then we have this automatic neuronal feedback that says, that doesn't taste sweet, savory, salty, like something I should consume. But culturally, if we know that this is, healthy and refined, then we learn, oh, I didn't puke when I ate that. And I actually liked it, so I'll do it again. But it takes training that interaction between our taste receptors and our brains. But again, those individuals, we have such a range of different taste receptors that different individuals produce, that what might be noxious to you is sweet and savory to the person sitting next to you, because of all the different receptors that are being produced because of their genes. Right.
And so, if we're talking about environment and taste, what about smokers? Do we know, is that affecting it the same way, or is that a different way?
Well, I think that there are many answers to that question, and that there are extreme tastes in a lot of cigarettes. There are a lot of additives. There are things that would activate taste receptors, and that could downregulate them, and that would be suppressing that need to say this doesn't taste good. And so you get this feedback, and so and this is very likely going to spread around into your other taste world, your perceptive world. If those sort of extreme chemicals are causing your receptors to downregulate themselves, so that might be how that's happening. Every one of our sensory systems has these ranges. Our hearing system has the same kind of genetic differences and environmental influences, right? Our touch systems, we have a range of thermosensory, things that's why some people are like, it's so cold in here, and other people are like, it's really hot. And part of that, there's part of it is thermoregulation with body mass and everything, but some of it has to do with your external receptors that are getting information from the environment. And how you perceive the world is different than how they do. And so my husband and I like to just have that argument done just by having thermometers around the house to say, it may feel like it's warm, but it's actually, So hearing and touch and smell, as I said, smell, we have hundreds of different receptors in our olfactory system, but not everybody has the same ones.
So just going back a little bit, back to the vision. When you show those images, some people see the face, some people see the face. And oftentimes, if you look at those images on the internet, you see that, if you have left hemisphere dominant, you'll see this. If you have right hemisphere dominant, you will see this. But is it true?
I have no data whatsoever on hemisphere dominance. I have no idea. I think that there are definitely data that we develop different specialties of things in different hemispheres in terms of, while you might have both sides being sensory or motor or even speech, you might have one that would be working for one aspect of that versus the other. And the brain is incredibly bilateral, but also the networking of the brain is so complex that I would never say absolutely no, never. There's no sort of thing as lateralization. Yeah, so, sorry. No answer for that one. Yes?
And I'm just wondering, is there any correlation between the different sense modalities? Like, let's say if, you have a high acuity for smelling, is that going to, are you going to be, highly sensitive to touch as well?
Right, so there
What's the
are...
question? I'm sorry, I didn't understand.
So the question is, sensory integration is kind of what you're asking. She's saying, if you have a high acuity for smell and touch, do you have, is this sort of additive, cumulative? Do you have things in your brains that help to put those together? And there definitely are places of our brains that integrate certain senses, especially smell and taste, right? And hearing and touch. We have our brains, our cortex, and well, actually, interestingly, are these things called the limbic system, and are these places that we even respond to fear and anxiety, have integrative points for different sensory systems as well. So, yeah, that's an excellent point. You rarely ever you experience one sensory modality. Like it's hard to ever, other than when you have a cold, you recognize that you can still have some taste, but you don't have smell as much, but everything tastes a little differently because those two modalities work so well together. But you also might lose some touch in your mouth, feeling about how things feel when they taste. So you've got a lot of touch feedback in your mouth as well. That kind of sensory. There are people that say, I just don't like how tomatoes feel in my mouth. And so, there's this integration of these. But, yeah, it is very difficult to fully separate full sensory experience from another, because you're always getting information about your thermal environment. You're always getting information about your light environment, unless you are being in some way sensory deprived, which is unusual. So perceptions are then constantly the sort of smash-up of all of those things happening together for global events. But Michelle's still going.
Oh no, I'm just thinking.
Okay.
So what is the final verdict on like the verminasal organ? Like, how cats and dogs have it. Do humans actually have it or not?
So the vomer nasal organ is this organ that has been detected in a lot of mammal species that detects pheromones. And pheromones are volatile, like chemical senses, like what we smell. And so the question is that what's going on with humans? We have one, but it's called vestigial, because we don't have any good data to suggest that it is actually working in the context, especially of mating, mate choice, and all of that. But there are still lots of great data on how we are able to detect volatile compounds that are considered pheromonal from our other human species. People are able to sniff out mates, more compatible mates, in a blind way. And so how else would they be able to do that? So there's this great study that's been replicated now over a dozen times, where guys will wear a t-shirt and night, and just well, that's all they have to do, and then they put the t-shirt in the bag, and then women come in and they smell the t-shirt and they rate the t-shirt of what's the most attractive, what do they like the most. And part of this has to do with being able to show that those women that chose those men's scents have very different genetics. So we are able to sense through some means that type of information. How that is actually happening, I have no clue. Yes?
So I'm interested in the sort of plasticity response of the environment, and if the jury seems to be on this, but if you think that there's a developmental window in which we can develop maybe things become more fixed, maybe not all things, but some things become fixed, or they become less likely to change over the lifespan of the organism. Humans, I guess.
Yeah.
I mean,
Well, I think it just depends on the actual modality. There's certainly critical periods for development of all of our senses, right? And if you have sensory deprivation, then it's very unlikely that you're going to be able to develop that. Development is crucial. But as one of my colleagues said, you know, it's all genetics and it's all environment. So, right. So, yes.
You were mentioning the smell test. There's actually dating sites which use that.
Send in your t-shirt.
Yeah, it's
Yes.
the 3 immunity factors that if you have completely separate, then you attract to the smell of the person. And supposedly when you're dating, it leads to enhanced experience dating, let's say. The thought is that back when we had a lot of plagues, or when we've gone through different series of plagues, that the Black Plague and the rest, that this would allow one of the 2 parents to survive the plague and raise the child. Because they had such different immunity, there's a better chance that one of the 2 would not die.
Well, the thing about it is that I would argue from the evolutionary standpoint that anything like that evolved way before any of that. And that, because we see it in other organisms. Mice can do this. Mice can differentially sniff out those with a better major histocompatibility complex. So it's
It could have been other infections from the past. Yeah.
could have been, it's a lot of evolution in that. Yeah. Carl.
Most reports of people who use LSD visual hallucination, but does that work primarily in the visual cortex? Does it affect all modalities, not the drugs that will give you primarily auditory hallucinations?
So the question is, drugs like LSD, what are they doing to your perceptual world, basically? I mean, is it because, so let me think if I'm getting your question right, are we changing how the stimulus energy is being transduced, coming in, or how our brain is perceiving that same information? And from what little I would understand of that, I would say it have to be how you're perceiving it. That you're not changing your actual, that transduction methodology. You're not changing how a cone works. But maybe, I don't know, it depends on how much LSD you take, I guess.
Although there is this phenomenon associated with LSD use where you get persistent visual snow, and actually, like, I've read a couple of studies which then link it to increased actual perceptual awareness. Like, they do different, like, masked stimuli, and people who develop that over time from LSD use or other hallucinogen use, they actually have higher sort of visual awareness of those masked stimuli.
But higher visual awareness, again, it probably goes back to your perception of your visual world and not the rods and cones changing how they are transducing light. Alex, do you have any?
Well, I was thinking looking at it, there's actually enough there's evidence to suggest that there would be a mechanism by which marijuana could changes the actual transduction of tone and raw signaling. So,
Well, they are neurons.
but that's not to say, but let's use a different question.
Yeah, I have no idea. Yeah.
So this is kind of going back to the development question. What about people who are born deaf or blind and then have those sort of senses restored several decades later? Like,
I don't really know that they don't have them. If there's any restoration, it's never been with the same level of acuity that you would have them from development. There's some great.
So do you know, I guess, like, I've heard of cochlear implants, but I don't really know how they work. Is that just in the brain, or is it about getting the signal transduced to the brain and then the brain has to figure it out?
So it's about, the cochlear implants are about getting the signal transduced to the brain. You actually absolutely have it correct.
Okay.
Now, what the brain does with that information depends on the point in development at which the individual gets the implant. So there is a critical period that is too far to get the same acuity, which is why they've been pushing to have those earlier and earlier, because those networks in your brain are being set, and without activity, without input from a sensory system, then the brain says, okay, well, I'm not getting this activity, so I will not be working on that. I will do something else, basically, because our brains are incredibly energetically energy hogs, right? So yes, you have to have that input, but still, right? There are some recordings that you probably can get them online. I have them for some classes that, like us seeing the visual world of somebody without cones, you can hear what that sort of input would be with a person with cochlear implants. So, and it's very different than what does a person who's developed hearing.
Even though we're hardwired for visual part of the brain, the oral part of the brain, we're hardwired for where we process things for the most part. If there's damage, of course, we can move, maybe move the areas if needed. They're finding that we have plasticity, the younger, the easier. And if from birth, if we're shut off from hearing, or if we're shut off from sight, our brain eventually, like when it goes through development without using those parts, it starts to use those parts for the senses you do have. That's why people who are blind from birth and get blind, from childhood or even later on, they eventually start to use those visual parts of the brain for hearing, and that's why they can tune and hear things and discriminate sounds much better than human lie detector in some ways. Just from hearing sounds, hearing people speak, they can process a lot.
I think that's a fascinating field. I know that there's a tremendous amount of plasticity that's going on, but I don't know these case studies, but that sounds really interesting.
The younger, the easier, like you said.
So this brings, there are a few instances of people who, when they hear music, for instance, they see the music. And I'm just wondering, is this in some way, does this light up when I hear, for instance, does it all of a sudden translate to my visual lobes as opposed to just my auditory lobes, or what happens in a case like that?
So this is the kind of the crux of the whole issue of perception, and that we never just use one part of our brain for anything, really. There's this mythology that has been out there. You only use 10% of your brain. That's not true. And so our brain is a network that's constantly networking to make sense of the world. And so if part of how you perceive and think of music includes how you can visualize that sound or what it means to you, then those connections are there, and it's going to be one of those things that, and every time that you have that perceptual event, it will actually strengthen those networks, and so that it becomes your norm. And so, and again, and everybody has their own norm for what their perceptual world is with certain stimuli.
I think with regard to we call that synopsis, yeah, and I think that people did fMRI studies on people who have that. For instance, some people will see colors when they're shown numbers, or
Oh, I see what you're saying. Yeah, that.
Yeah.
Oh, I thought it was something else you were talking about. Yeah.
and I think that when they do fMRIs, they actually do see activation of the. For instance, if they're shown numbers and they say there's a blue 5 and there's a black 5, it does turn on their visual cortex.
But the fact is that.
Aesthetic.
But the problem with this concept of only turning on one area of the brain is, again, this whole.
Right, just relative to the control group that doesn't have synesthesia, you might see elevated levels of activity in the visual cortex when you're shown just, when you're shown an not a visual stimulus, whereas, we would see less control group. But of course, yeah, there's activation.
All right. Are we done?
This may be a good point to
Okay.
stop.
Thank you so much, everyone, for your